Good day, everyone. Welcome to our first topic for the MLS 411 leg. And I am Sirab. I will be your lecturer for today's video. Microorganisms and their activities are vitally important to virtually all processes on Earth. Microorganisms matter because they affect every aspect of our lives. They are in us, on us, and around us. And the lesson objectives of this study are the following. So first is to describe the historical development of microbiology. Next is to enumerate the development of science with emphasis on persons, on scientists, and their contributions. Next is to explain the divisions of microbiology. And lastly, to define commonly used terms in microbiology. Now let us proceed to the etiology of the word microbiology. So it came from these three words, micros meaning small, bios meaning life, and logos meaning to study. Technically, a microorganism or a microbe is an organism that is microscopic. So the study of microorganisms is called microbiology. It is the study of all living organisms that are too small to be visible with the naked eye. This includes bacteria, archaea, viruses, fungi, prions, protozoa, and algae, collectively known as microbes. So these microbes play key roles in nutrient cycling, biodegradation or biodeterioration, climate change, food spoilage, the cause and control of disease, and biotechnology. Thanks to their versatility, microbes can be put to work in many ways, so making life-saving drugs, the manufacture of biofuels, cleaning up pollution, and producing or processing food and drink. What comes to your mind when you hear the words bacteria or virus? Yes, it can be the words infection or disease. So there's a difference between infection and disease. Infection, often the first step, occurs when bacteria, viruses, or other microbes that cause diseases enter your body and begin to multiply. Disease occurs when the cells in your body are damaged as a result of the infection and signs and symptoms of an illness appear. In response to infection, your immune system springs into action. An army of white blood cells, antibodies, and other mechanisms goes to work to rid your body of whatever is causing the infection. For instance, in fighting off the common cold, your body might react with fever, coughing, and sneezing. And way back before, there are pathogens that almost swept the whole of human race. They caused this, what we call the epidemics. So the causative agent of plague is the Yersinia pestis, cholera. Causative agent is Vibrio cholerae, and smallpox is the variola virus. Now let us differentiate a bacteria and a virus. Bacteria, their genetic material is not enclosed in a special nuclear membrane. Bacterial cells are called prokaryotes from the Greek words meaning pre-nucleus. And bacteria are relatively simple and they are single-celled organisms. While viruses are so small that most can be seen only with an electron microscope, and they are acellular or not cellular. So the virus, the core is surrounded by a protein coat which is sometimes encased by a lipid membrane called an envelope. And a virus core is made up of only one type of a nucleic acid. It can be either a DNA or an RNA, but not both. Now I have a question for you. Why do we study microbiology? So these are the beneficial aspects of microbiology. So the normal microbiota provides a first line of defense against microbial pathogens. So it assists in digestion, play a role in toxin degradation, and contribute to maturation of the immune system. Decomposers make vital nutrients available to an ecosystem's primary producers, usually plants and algae. So decomposers make vital nutrients available to an ecosystem's primary producers, usually plants and algae. So bacteria are used in industry in a number of ways that generally exploit their natural metabolic capabilities. They are used in manufacture of foods, in production of antibiotics, probiotics, drugs, vaccines, starter cultures, insecticides, enzymes, fuels, and solvents. And pathogens or opportunistic pathogens cause disease when the host's defenses are compromised or when they grow in part of the body that is not natural to them. Alright, so after knowing all of those basic principles of microbiology, let us proceed to the various scope of microbiology. So the first one is general. So the purpose is to study and classify the different microbes. Medical microbiology, so pathogens, diseases they cause, and the body's defenses against diseases. And veterinary microbiology, it is the study of the spread and control of diseases in animals. We also have agricultural microbiology. The role is in the microbes in plants and livestock. And then next is sanitary microbiology. The role is for the processing and disposal of garbage and sewage waste, purification and processing of water. We also have industrial microbiology, which is the function is for the production of beer, wine, alcohol, vitamins, antibiotics, etc. We also have microbial physiology and genetics. So it is the study of the genetic manipulation, and then environmental is the study of the cycling and recycling of elements by microbial, environmental, and geochemical processes. Now let's proceed to the historical developments of microbiology. So this is the beginning. So bacterial ancestors were the first living cells to appear on Earth. So for most of human history, people knew little about the true causes, transmission, and effective treatment of disease. Let's look now at some key developments in microbiology that have spurred the field to its current technological state. So the first time the word cell was used to refer to these tiny units of life was in 1665 by a British scientist named Robert Hooke. Hooke was one of the earliest scientists to study living things under a microscope. So the microscopes of his day were not very strong, but Hooke was still able to make an important discovery. When he looked at a thin slice of cork under his microscope, he was surprised to see what looked like a honeycomb. Dutch merchant and amateur scientist Anton van Leeuwenhoek was probably the first to observe live microorganisms through the magnifying lenses of more than 400 microscopes he constructed. So between 1673 and 1723, he wrote about the animalcules he saw through his simple single-lens microscopes. Van Leeuwenhoek made detailed drawings of an organism or of organisms he found in the rainwater, feces, and material scraped from teeth. These drawings have been since these drawings have since been identified as representations of bacteria and protozoa, as you can see in the picture on the right. So that was Sir Robert Hooke, and the picture on the lower right of the presentation is Sir Anton van Leeuwenhoek with his single lens microscope. Now let's proceed to the transition of the historical developments of microbiology. So in 1700s, the focus was plant and animal life and the attempts for categorization of microbes. Carlos Linnaeus is the father of taxonomy, which is the system of classifying and naming organisms. One of his contributions was the development of a hierarchical system of classification of nature. Today, this system includes 8 taxa. So, first is the domain, next is kingdom, then phylum, then class, order, family, genus, and species. So that's why binomial nomenclature, it is a system of nomenclature in which each species of animal or plant receives a name of two terms, of which the first identifies the genus to which it belongs, and the second, the species itself. So the genus is the generic name, whereas the species is the specific name in a binomial nomenclature. So it is printed in italics or underlined in script. Just like, for example, Streptococcus pneumoniae. Next, we have the theory of biogenesis and abiogenesis. So let's differentiate the two. So abiogenesis is an ancient belief about the origin of life. So this is also known as the theory of spontaneous generation of life. So the theory of abiogenesis stated that the origin of the living organisms is due to non-living substances, or it is a spontaneous incident. However, until now, scientists have been unable to accomplish this theory by experiments. While biogenesis, on the other hand, is the currently accepted theory regarding the origin of a new life. So the theory of biogenesis states that the origin of life is because of pre-existing living cells or an organism. So Louis Pasteur, Francesco Redi, and Lazarus Spallanzani experimentally proved this theory. Next, there are two German scientists, Theodor Schwann and Matthias Jacob Leiden. Theodor Schwann is a zoologist. Matthias Jacob Leiden is a botanist. Suggested that cells were the basic unit of structure and function of all living things. So later, in 1858, the German doctor Rudolf Virchow observed that cells divide to produce more cells. He proposed that all cells arise only from other cells. The collective observations of all three sciences from the cell theory, which states that all organisms are made up of one or more cells, all the life functions of an organism occur within cells, and all cells come from pre-existing cells. And it was also followed by Sir Louis Pasteur in 1864. So way back in 1858, Rudolf Virchow challenged the case for spontaneous generation with the concept of biogenesis, hypothesizing that living cells arise only from pre-existing living cells. Because he could offer no scientific proof, arguments about spontaneous generation continued until 1861, when the issue was finally resolved by the French scientist Louis Pasteur. So, Sir Louis Pasteur questioned, is there a life force in air that can cause microbes to develop by spontaneous generation? And is there a means of allowing air to enter a container but not the bacteria that are present in it? So that's why Louis Pasteur prepared a nutrient broth similar to the broth one would use in soup. Next, he placed equal amounts of the broth into two long-necked flasks. He left one flask with a straight neck, the other he bent to form an S shape. Then he boiled the broth in each flask to kill any living matter in the liquid. The sterile broths were then left to sit at room temperature and exposed to the air in their open mouth flasks. After several weeks, Pasteur observed that the broth in the straight neck flask was discolored and cloudy, while the broth in the curved necked flask had not changed. He concluded that germs in the air were able to fall unobstructed down the straight necked flask and contaminate the broth. The other flask, however, trapped germs in its curved neck, preventing them from reaching the broth, which never changed color or became cloudy. So the conclusion for this experiment of Sir Louis Pasteur is that germs come from other germs and do not spontaneously generate. So if spontaneous generation had been a real phenomenon, Pasteur argued the broth in the curved neck flask would have eventually become reinfected because the germs would have spontaneously generated. But the curved neck flask never became infected, indicating that the germs could only come from other germs. And after that, so the period from 1857 to 1914 has been appropriately named the Golden Age of Microbiology. So rapid advances, spearheaded mainly by Pasteur and Robert Koch, led to the establishment of microbiology. Discoveries included both the agents of many diseases and the role of immunity in preventing and curing disease. One of the key steps that established the relationship between microorganisms and disease occurred when a group of French merchants asked Pasteur to find out why wine and beer soured. They hoped to develop a method that would prevent spoilage when those beverages were shipped long distances. At the time, many scientists believed that air converted the sugars in these fluids into alcohol. And that's why Pasteur's work proved in which microbes are responsible for fermentation and the spoilage of food. Vinegar is produced when bacteria ferments ethanol in wine. Spoilage bacteria could be killed through pasteurization. And pasteurization, by definition, is the process of heating the object, whatever it is. For example, milk, it is heated, then the harmful things are killed and made safe. While fermentation, on the other hand, is the chemical breakdown of a substance by bacteria, yeasts, or other microorganisms, typically involving effervescence and the giving off of heat. So before the time of Pasteur, effective treatments for many diseases were discovered by trial and error, but the causes of the diseases were unknown. Germ theory of disease is based on the concept that many diseases are caused by infections with microorganisms, typically only visualized under high magnification. Such microorganisms can consist of bacterial, viral, fungal, or protist species. Although the growth and productive replication of microorganisms are the cause of disease, environmental and genetic factors may predispose a host or influence the severity of the infection. For example, in a host that is immunocompromised due to AIDS or old age, An infection may result in more severe outcomes than in individuals who are fully immunocompetent. The notion that diseases could be spread by seed-like entities was first described in the 1500s by Girolamo Fracastoro and were categorized based on how they could be transmitted. Later, Agostino Bassi in the early 1800s conducted a series of experiments which demonstrated that a disease afflicting silkworms at the time was caused by a parasite. Then Bassi theorized that disease in humans and animals was also caused by microorganisms. Bassi's work served to influence Louis Pasteur, who is accredited with the germ theory of disease following his experiments demonstrating the relationship between microorganisms and disease. So in 1865, Louis Pasteur demonstrated that life did not arrive spontaneously from non-living matter, and he discovered that another silkworm disease caused by a protozoan. So in 1840s, Ignace Semmelweis advocated hand washing to prevent spread of puerperal fever. And Joseph Lister performed surgery under aseptic conditions using phenol, and he proved that microbes cause surgical wound infections. And lastly, Robert Koch established experimental steps for directly linking a specific microbe to a specific disease. And now let's proceed to the microbial etiology of important diseases. Established Koch. So for the Vibrio cholerae, it is the causative agent of cholera. And then Mycobacterium tuberculosis, causative agent of TB or tuberculosis. And Bacillus anthracis is the causative agent of anthrax. And now let's proceed and discuss to the Koch postulate. So Koch established Koch postulates a sequence of experimental steps for directly relating a specific microbe to a specific disease during the past 100 years. So these same criteria have been invaluable in investigations, proving that specific microorganisms cause many diseases. So developed in the 19th century, Robert Koch postulates are the 4 criteria designed to assess whether a microorganism causes a disease. So as originally stated, the 4 criteria are: first, the microorganism must be found in diseased but not in healthy individuals. Second, the microorganism must be cultured from the diseased individual. Third, inoculation of a healthy individual with a cultured microorganism must recapitulated the disease. And finally, the fourth, the microorganism must be isolated from the inoculated. So diseased individual and matched to the original microorganism. Koch's postulates have been critically important in establishing the criteria whereby the specific community agrees that a microorganism causes a disease. But there are also exceptions to Koch postulates. So one of which is that many healthy people carry pathogens but do not exhibit symptoms of the disease. Second, some microbes are very difficult or impossible to grow on artificial media. Third, to induce a disease from a pure culture, the experimental animal must be susceptible to the pathogen. And lastly, certain diseases develop only when an opportunistic pathogen invades a weakened host. Often, a treatment or preventive procedure is developed before scientists know why it works. So the smallpox vaccine is an example. Edward Jenner is considered the founder of vaccinology in the West in the year 1796 after he inoculated a 13-year-old boy with vaccinia virus or the cowpox and demonstrated immunity in smallpox. In the year 1798, the first smallpox vaccine was developed. Over the 18th and 19th centuries, systematic implementation of mass smallpox immunization culminated in its global eradication in 1979. After the relationship between microorganisms and disease was established, medical microbiologists next focused on the search for substances that could destroy pathogenic microorganisms without damaging the infected animal or human. And chemotherapy is a drug treatment that uses powerful chemicals to kill fast-growing cells in your body. Chemotherapy is most often used to treat cancer, since cancer cells grow and multiply much more quickly than most cells in the body. Many different chemotherapy drugs are available, such as our quinine. So it came from a tree bark, so it is used for treating malaria. Paul Ehrlich was the imaginative thinker who fired the first shot in the chemotherapy revolution. So as a medical student, Ehrlich speculated about a magic bullet that could hunt down and destroy a pathogen without harming the infected host. And in the year 1928, the first antibiotic was discovered by accident. Alexander Fleming, a Scottish physician and bacteriologist, almost tossed out some culture plates that had been contaminated by mold. Fortunately, he noticed the curious pattern of growth on the plates. A clear area where bacterial growth had been inhibited encircled the mold. And the first antibiotic, penicillin, came from Penicillium notatum, and later it was renamed as Penicillium chrysogenum. In the year 1930s, sulfonamides were synthesized, and in the 1940s, penicillin was tested clinically and mass-produced. So the quest to solve drug resistance, identify viruses, and develop vaccines requires sophisticated research techniques and correlated studies that were never dreamed of in the days of Koch and Pasteur. The groundwork laid during the golden age of microbiology provided the basis for several monumental achievements in the years following. So here are the modern developments of microbiology. Microorganisms can now be genetically modified to manufacture large amounts of human hormones and other urgently needed medical substances. In the 1960s, Paul Berg showed that fragments of human or animal DNA, or the genes that code for important proteins, can be attached to a bacterial DNA. The resulting hybrid was the first example of recombinant DNA. Recombinant DNA, or small r, capital DNA, technology inserts recombinant DNA into bacteria or other microbes to make large quantities of a desired protein. So this field combines elements from two other areas of study, including microbial genetics, which studies the mechanism by which the microorganisms inherit traits, and molecular biology, which looks at how genetic information is carried in molecules of DNA and how DNA directs the synthesis of proteins. Now, let's proceed to microbes and human disease. We all live from birth until death in a world filled with microbes, and we all have a variety of microorganisms on and inside our bodies. The normal microbiota not only do us no harm, but in some cases can actually benefit us. For example, some normal microbiota protect us against disease by preventing the overgrowth of harmful microbes, and others produce useful substances such as vitamin K and some B vitamins. So resistance, this term is defined as the ability to ward off disease factors. So, such as our skin, stomach acid, antimicrobial chemicals. And infectious disease, so when a pathogen overcomes the host resistance, so disease results. Next, we are down to the emerging infectious diseases. So these recent outbreaks point to the fact that infectious diseases are not disappearing but rather seem to be re-emerging and increasing. In addition, so a number of new diseases, emerging infectious diseases or EIDs, have cropped up in recent years. So emerging infectious diseases, so these are diseases that are new or changing and are increasing or have the potential to increase in incidence in the near future. So example of our emerging infectious diseases, so we have severe acute respiratory syndrome or SARS is an emerging infectious disease that first appeared in China in 2002. It is a viral infection caused by the SARS-associated coronavirus or the SARS-CoV. And we also have H1N1 influenza or the flu. So H1N1 influenza flu is also known as the swine flu. It is a type of influenza caused by a new virus called influenza H1N1. And H1N1 was first detected in the United States in 2009. And that same year, the World Health Organization or the WHO declared H1N1 flu to be a pandemic disease, a disease that affects large numbers of individuals in a short period of time and occurs worldwide, just like COVID-19. And lastly, we have avian influenza A or H5N1 or the bird flu. H5N1 caught the attention of the public in the year 2003 when it killed millions of poultry and 24 people in Southeast Asia. Avian influenza viruses occur in birds worldwide, and in 2013, a different avian influenza H7N9 sickened 131 people in China. And these are the various references used for this topic. Thank you so much for listening, everyone, and have a good day.